Hello everyone, myself P.S.Patil, working as Assistant Professor in KRT Arts, BH Commerce and AM Science College Nasik. I am welcome you in our ninth lecture. In this lecture, we are going to discuss the characteristics values of chromatogram. In the previous lecture, student, we have seen the technique of chromatography, what is chromatogram, and the development of that chromatogram by paper chromatography as well as column chromatography. This part we have already seen in the 8th lecture in the previous lecture, right? In the today's lecture, we discuss the characteristics values of chromatogram. So, this is the content of the today's lecture, characteristics values of chromatogram. There are different characteristics value, so we will discuss that characteristics in the next slide. So the characteristics values of chromatogram, these are the 6 different characteristics values of chromatogram. The first one is the chromatogram. It is denoted by letter TR, retention time capital R, capital TR, dead time TM, mole fraction Rx, retention factor K, separation factor that is alpha. Okay, so one by one we are going to discuss the characteristics value of chromatogram. The first is the chromatogram. In the previous lecture, we have seen this concept chromatogram. Chromatogram, it is nothing but a column or a paper strip on which we introduce the sample, right? That is called spotting, and we put that paper into the mobile phase. Okay, so what happened, that mobile phase, it is running on that paper or running on that stationary phase, and the separation of the component takes place according to the adsorption phenomena. That is nothing but the chromatogram, or in another word we simply say that it is a graphical representation of detector response versus time. So this chromatogram is a two-dimensional plot with the ordinary axis giving a concentration in terms of detector response versus time. Okay, the detector gives the response as a peak whose height should be ideally depend on the concentration of a particular component. Suppose we are getting a very sharp peak, that means the concentration is very less. Okay, and if we are getting a broad peak, that means the concentration is also more of that compound. Okay, chromatogram is the time required from the injection of sample to the elution of the last component, right? So student, in the elution analysis, in the previous lecture we discussed this elution analysis. So in that analysis we are taking one column, we introduce the sample at the top of the column. That sample contain A, B, C component, right? So there is a elution of A component first, then B and last C. That means the time required to elute that last component, that is C, from the column, that is nothing but the chromatogram. Okay, so this is the one chromatogram is there. This is detector response versus time. It is a graphical representation of detector response versus time. Okay, chromatogram, it is nothing but a graphical representation of detector response versus time. Here we inject a sample. So here the injection of the sample. Then this is the TM, that is called dead time. Time required to remove the mobile phase, that is nothing but the TM, that is dead time. And time required to remove the first component elution, that is the retention time. Okay, if the component contain sample contain 3 component, then we say that TR1, TR2 and TR3. Okay, so depending on the component present in the sample, this retention time is there. Okay, how the width of this peak is calculated? We are drawing the tangent like this. We are drawing the tangent like this and we are finding the width of this peak. Okay, this is nothing but the chromatogram. Then the next is the retention time. Retention time is the time that a solute spent in the column or it can be defined as solute spent time in the stationary and the mobile phase. The longer the retention time depend on the interaction of analyte with the stationary phase. Stronger the interaction, more will be the retention time, or more will be the interaction time. So here also the previous figure is there. Injection takes place. This is the date time. This one is the retention time. This W is the width of this peak. Detector response versus a time. This TR, it is the retention time of the solute. W is the width. TM is the date time or time for all solute in the mobile phase. This TR, it is the time in a stationary phase and time in a mobile phase. So TR, retention time, is equal to summation of TS plus TM. Okay. Dead time, it is denoted by letter TM, that is time required for the illusion of mobile phase is called a dead time. Means we are going to take a column which is filled with the stationary phase and we introduce the mobile phase. So the time required to elude that mobile phase, it is nothing but the dead time. It is calculated by this two way. U is equal to L upon Tm. L is the length of the column, U is the velocity of the column and Tm is the dead time. Then another way we are find out the retention time also. V is equal to L upon Tr. V is the velocity of sample, Tr is the retention time and L is the length of the column. So by this we find the date time and the retention time. Okay. Then the next is mole fraction. It is denoted by letter Rx. So by using this formula, we are find out the mole fraction. CM/Vm upon CM/Vm plus CS/Vs. Okay. The Rx, it is the mole fraction. Cm is concentration of solute in mobile phase. Vm is the volume of mobile phase. CS is the concentration of solute in the stationary. So S stand for stationary and M stand for the mobile phase. Vs is the volume of the stationary phase. Here, if the Rx value it is 0, then what it indicate? Then the solute is strongly adsorbed on the stationary phase and there is no separation. Okay, that means if there is no Rx value, means it is 0, then that component it is strongly adsorbed on the stationary phase. It never affected by any mobile phase. It is strongly adsorbed on the stationary phase. So there is no separation. Okay. If the Rx is 1, then the mobile phase and the solute travel at the same rate and there is also no separation. Okay. That means mobile phase and the component, they are travel at the same rate. So we are not getting the separation in the column. The ideal value of this Rx, it is in between 0 to 1. Then the separation occur and that separation it is called a good separation. Okay. So this is the mole fraction. Then the next is the retention factor. Retention factor, it is denoted by a letter small k. This retention factor is the time the component interact with the stationary phase relative to the time the component spent in the mobile phase. That means the component interact with the mobile stationary phase and the component which spent in the mobile phase, that is nothing but the retention factor. If the mobile phase flow rate it is double, if we pass the mobile phase in the double rate, its flow rate is double, then both the retention time and the dead times are also double. Okay. However, the retention factor values remains unchanged. Either it is double or it is remain unchanged. Okay. When the component is injected into a column, then its total mass it is divided into two quantities, the mass in the mobile phase and the mass in the stationary phase. Okay, means we introduce the sample, it is divided into two categories, the mass which is present in the stationary phase and mass which is present in the mobile phase. The values of these quantities are depend on total mass and the retention factor, but in their ratio the retention factor is a constant. K is equal to ms upon mm. The factor K, which is independent of the flow rate and the length of the column. This factor it is independent on length of the column as well as the flow rate of the mobile phase. That can vary with the experimental condition. K is the most important parameter in the chromatography for determining the behavior of the column. And the value of K should not be too high, otherwise the time of analysis is also elongated. Then the last factor, that is the separation factor, it is denoted by letter alpha. This separation factor, it is nothing but the ability of given stationary phase to separate the two components. Ability of the stationary phase to separate the two components, that is nothing but the separation factor. This parameter used to describe how well two solutes are separated by the chromatographic system. How two solutes are separated by the chromatographic system, that can be shown by this parameter, that is separation factor. It is denoted by formula alpha is equal to K2 upon K1. This K1 it is the capacity factor of the first solute and K2 is the capacity factor of the second solute. Okay. So these are the 6 parameters of this chromatogram. In the next lecture, we are going to discuss the theories of chromatography. In that we discuss plate theory and the rate theory. Okay student, thank you.